Hello, interested everyone. Welcome to class 12 biology. In our previous video, we have been discussing about microbes which are used in household products and industrial products. We have seen how microbes are involved in production of curd, cheese, idli and dosa, and different types of drinks. Right? We have also seen microbes which are useful in production of antibiotics, chemicals and enzymes, and then there are also microbes which are involved in production of bioactive molecules like cyclosporins and statins. In this video, we'll be discussing about microbes which are used in sewage treatment and microbes in production of fuel called biogas. So the first one is microbes in sewage treatment. What do we mean by sewage and why are the sewage treatment plants necessary? So by sewage, we mean municipal wastewater. We use water for different purposes in our day-to-day life. We use water in the kitchen, we use water in bathroom and toilets, right? So even big factories, they use water for different purposes during their production process. So these water, after they are being used, they become wastewater. A small household containing a small family produces a large amount of wastewater every day. Now imagine a big town or a big city which contains a large number of such families. It will produce huge amount of wastewater every day, and that wastewater is called as municipal wastewater or sewage. Now this municipal wastewater or sewage ultimately ends up being dumped into natural water bodies like oceans, rivers, lakes and ponds. Now ideally, the sewage or municipal wastewater, they should not be directly released into natural water bodies because these contain human excreta. So the human excreta contains pathogenic microbes and other organic matters. So if we release sewage directly into natural water bodies, it will contaminate the water and it will also pollute the water, which will make the natural water bodies unusable for future purposes. So therefore, this municipal wastewater or sewage, they should not be directly released into the water bodies. They need to be treated first. Right? Before being released into natural water bodies, the sewage should be treated first in sewage treatment plants. So in sewage treatment plants, these municipal wastewater, they will be treated with heterotrophic microorganisms. So these heterotrophic microorganisms, they will make the sewage water safe to be released into the natural water bodies. And that's why sewage treatment plants are necessary. So the sewage treatment process involves two stages, right? Primary treatment and secondary treatment. So during the primary treatment stage, large and small solid particles which are present inside the sewage will be physically removed through filtration and sedimentation processes. So those solid particles which are floating inside the sewage, they will be first of all removed through sequential filtration process, and those solid materials which are not removed through filtration process, they will be removed through sedimentation process, during which the wastewater will be allowed to stand in large containers, right, in large tanks, and under the influence of gravity, the solids will be allowed to settle down at the bottom. That is called a sedimentation process. And the solids that settle in the tanks, they form primary sludge, which is removed, and the supernatant, that forms the effluent, and effluent is taken for secondary treatment, which means the next stage. In this particular diagram, I have tried to show you the primary treatment process. Imagine this container is really large, a large tank, right, in which the sewage, the raw sewage water is collected. Right? So the raw sewage water is collected in these large tanks, and the floating debris or the solid particles which are floating, they will be removed through filtration process, and the other solid particles which are not being removed through filtration process, they will be allowed to be removed by sedimentation process, during which under the influence of gravity, the solid particles will settle at the bottom. That will form the primary sludge. The primary sludge will be removed, and the effluent, that is taken for secondary treatment, because this is still not safe to be released into natural water bodies. The effluent is considered unsafe to be released into water bodies because it still contains a large amount of dissolved organic matter in it. The dissolved organic matter, they are pollutants, they can pollute the water bodies. So therefore, effluent is not released into water bodies, it is taken for secondary treatment process. So during the secondary treatment process, the organic matter present inside the effluent will be removed so that it becomes safe to be released into water bodies. Now, throughout the primary treatment process, we haven't seen the role of microorganisms. what we have seen is we have seen the removal of large and small solid particles inside the sewage through filtration and sedimentation process. These two are physical processes. There is no role of microorganisms in the primary treatment process. The role of microorganisms will be seen in the secondary treatment process, during which the microorganisms, they will help in the removal of organic matter present inside the effluent. During secondary treatment process, the effluent is taken in large aeration tanks. These aeration tanks allows the growth of aerobic microbes into flocs, which is nothing but masses of bacteria associated with fungus filaments to form mesh-like structures, and these mesh-like structures are called as flocs. Right. So the useful aerobic microbes, they consume the organic matter present inside the effluent, and it reduces the BOD. Now this is important. BOD stands for biochemical oxygen demand. It is the amount of oxygen required by the microbes to break down all the organic matter present in a liter of water. So BOD indicates the polluting potential of a water. If the water body contains a large amount of organic matter, it will show higher BOD. If a water body contains a low amount of organic matter, it will show lower BOD. So since aerobic microbes, they consume all the organic matter present inside the effluent, it reduces the BOD of the effluent, thereby reducing the polluting potential of the effluent. Now the effluent is safe to be released into water bodies. But before that, the effluent is allowed to settle, right? During which these masses of aerobic microbes, which forms flocs, they are allowed to settle at the bottom of the tank through sedimentation process, and it forms activated sludge, right? The activated sludge contains these microorganisms, right? So a small amount of activated sludge is kept as inoculum for the next batch of sewage treatment, right? And the remaining activated sludge is pumped into another container called aerobic sludge digesters, where anaerobic bacteria will be used to digest the sludge and produce biogas in the process, another useful process. Here in this diagram, I have tried to show you the secondary treatment process. So let's say this is the aeration tank, right, in which we have collected the effluent, which contains large amount of organic matter. With the help of air pump and this rotor, oxygen supply is provided for the aerobic microorganisms which grow within the aeration tank. Right. So these aerobic microbes, they will consume all the organic matter present inside the effluent, thereby bringing down the biochemical oxygen demand. So lower biochemical oxygen demand will signify less polluting potential of the effluent. So now the effluent is safe to be released into the water bodies. But before that, the effluent is allowed to settle, right? Under the influence of gravity, the activated sludge is formed at the bottom. A small portion of activated sludge is kept for the next use because it contains aerobic microorganisms in it, right? And the large portion of activated sludge is taken into another container called anaerobic sludge digesters, where anaerobic bacteria will digest the sludge and produce biogas. So here is a picture of aeration tank given in your textbook, and here is a picture of an aerial view of sewage treatment plant. And there is one significant statement made in your textbook regarding the usefulness of microorganisms in treatment of sewage. It says that till date, no man-made technology has been able to rival the microbial treatment of sewage. Right? We haven't been able to develop a technology which can clean the sewage or municipal wastewater as good as these microbes can do. Right? So this is quite a significant statement which shows the usefulness of microorganisms in sewage treatment. With this, we are done with microbes in sewage treatment. Before we move on to microbes in production of biogas, you may want to look up on some information regarding Ganga Action Plan and Yamuna Action Plan. Right. So these two action plans, they were initiated by Ministry of Environment and Forests in order to clean Ganga and Yamuna rivers. Right. Not much information is provided in your textbook apart from just mentioning their names, right? So it is advisable that you do a little bit of research and find some more information regarding these two action plans. Let us now discuss a little bit about microbes in production of biogas. We will see what is biogas, which microbes are responsible for the production of biogas, and how can we produce biogas with the help of these microbes. Biogas is a mixture of gases produced by microbes which mainly contains methane. So methane is the inflammable gas which makes biogas useful for fuel. Right. So biogas is a mixture of gases produced by microbes which mainly contains methane. And the microbes which are responsible for the production of biogas, they are usually bacteria. Right. So these are anaerobic bacteria which can perform anaerobic respiration, and during anaerobic respiration these bacteria, they produce methane gas along with carbon dioxide and hydrogen gas. Right. So therefore these bacteria, they are called as methanogens. So methanogens are a group of bacteria which produce methane gas along with carbon dioxide and hydrogen during anaerobic respiration. Right. One such example, one common example is methanobacterium. Right. And this methanobacterium is said to be used in anaerobic sludge digester during sewage treatment, right, which is used for production of biogas. We already said that. Right. Now such bacteria, they are also naturally found inside the rumen of cattle. Rumen of cattle means inside the gut of the cattle. Right. And these bacteria, they help them in digesting cellulose present inside the grass. Right. Till date, there is no known animal which can produce enzymes which can digest cellulose. Even cattle, they themselves do not produce any enzymes to digest cellulose. The bacteria present inside the rumen of the cattle, like these methanogens, they help in the digestion of cellulose. So therefore, the gobar or the cow dung released by the dungs released by the cattle, they already contain methanogens. Right. So in the production of biogas by the humans, we use gobar. So therefore, it is also called as gobar gas. Now let us see how we can produce biogas or gobar gas with the help of these microorganisms. In order to do that, we need to understand basic structures of a biogas plant. So here is the diagram representing a typical biogas plant. A typical biogas plant contains a concrete tank which is about 10 to 15 feet deep, and that concrete tank is filled with a slurry of dung through this inlet. So the slurry of dung becomes the source of methanogens. Right. So along with the slurry of dung, we can also add kitchen waste and other organic matter as well. Right. So the methanogen present inside the dung, it will anaerobically break down all the organic matter present inside the slurry, and it will produce biogas which consists of methane, carbon dioxide, hydrogen, etc. So this biogas, it is collected within a floating cover on the top. So let's say this is the concrete tank which is about 10 to 15 feet deep. In this concrete tank, we have the slurry of cow dung, right? So slurry of dung. On the top of this concrete tank, we put a floating cover, like a cover. Okay, so what this cover will do is this cover will collect all the biogas produced by the methanogens. And when this cover gets filled up with gas, it floats up, right? It raises up. Through that way, we know that there is a significant amount of biogas collected within it. And when a significant amount of biogas is collected within the floating cover, we can remove the gas or take the gas out through the outlet. Right? And this particular biogas can be used for different purposes, like biogas can be used for cooking and lighting as well. And after all the organic matter gets consumed by the methanogens through anaerobic respiration, the spent slurry can be used as fertilizer in the field. Right? So along with production of biogas, we also get fertilizer as well. That is about the basic structure of biogas plant, along with how we can produce biogas with the help of microorganisms. Right. So here is a model showing the biogas plant. Right. So this particular container acts like a biogas plant, and in this one you can see that we can mix different kinds of organic waste materials. Right. And inside this particular tank, this is called as the digester tank, in which the organic matter along with the kitchen waste matter, it gets decomposed or anaerobically broken down with the help of methanogens to produce biogas, which is collected within this gas holding tank. Right? And after the slurry is spent, it can be taken out to be used as manure or fertilizer. Right? And from this gas holding tank, we can take out the gas to be used in kitchen for cooking or for lighting as well. So this is a model of a biogas plant. So in the next video, we will be discussing about microbes as biocontrol agent and microbes as biofertilizers. So please read about this one before you watch the next video. Thank you.